Swayam Prabha. Digital India. Educated India.
Welcome to the organ printing course. So in this lecture, we'll discuss how we can bioprint a liver. Before that, we'll discuss what is the motivation of printing a liver tissue. So let's start with the motivation. So if you see, liver is a very important organ in our body. It does more than 500 functions. So that including some metabolic, immunologic, and detoxification processes it does in our body. So whatever the food we intake that goes to the from the gut under after digestion goes to the liver for metabolism. Further, liver also perform various immunological function and detoxification processes in our body. And liver accounts for almost like 2 to 5% of the body weight, and it is the largest solid organ in the body in that sense. And there are many patients who has been undergoing or waiting for transplants because of some end stage liver diseases like liver cirrhosis or liver fibrosis at the thing. Because due to these problems, the liver uh, they undergo liver transplantation where the whole liver is thinking. And if you see the liver tissue, it comprises of mainly hepatocytes, some somal cells, Kupffer cells, blood vessel associated cells. So different types of cells are present also in the liver tissue. So for bioprinting, we need to understand what are the different cell types and how they are arranged in the liver tissue. And these cells are arranged into a tissue structures with bile ducts and blood vessels. Liver has a very typical structures where the blood vessels and the bile duct they form a portal triad where they unless they pass so they come to this thing to the end of this a liver lobule is the structural unit of the liver, where in the liver lobule there are sinusoid-like structures that is made up of made up of this bile duct, blood vessels, this thing, and then and the liver cells like hepatocytes and hepatocytes sitting on the other side of the blood vessels, and the cholangiocytes they are sitting on the other sides of this bile duct. So they when the blood flowing through the liver, then these cells the hepatocytes they get oxygen, and due to this the oxygen gradient generally goes down along the path of this sinusoids. So this structure is called acinus. So that's hepatic acinuses, that is also the functional unit responsible for filtering toxins and blood and producing bile. So liver has a typical complex structure, so we need to reproduce that while doing bioprinting. But another important thing is liver has some self regeneration ability, right? So that if there is suppose there is some tissue is lost, if there is some tissue is damaged or diseased, that part if that part is resected out, liver has the ability to regenerate up to two-thirds of its damages that can be restored by itself. Uh, but when that if the liver the damage stuff is more than that, then in that case we need to intervene and we need to do certain treatment options are required. So one treatment options like several diseases include cell transplantation and extracorporeal devices are used for certain severe liver diseases with some amount of success. But whole orthotopic liver or whole or partial liver transplantation that remains the gold standard for liver failure, because in case of the whole liver failure or more than suppose the liver if the damage is more than two-thirds of the liver, then then the liver cannot regenerate by itself, and that time the transplantation is the main thing. But as with other tissue also we have discussed, the transplantation is not, because for transplantation we need to have a healthy liver tissue donor. But as as we have already discussed before also, there is a huge difference between the demand and supply. So that creates so transplants are not easily available for the needy people. Apart from that, high healthcare expenses is another another challenging thing for liver transplant, because the process is highly expensive, and this the there's a huge cost involved in that. Also, there is a risk of immune rejection, because we need to check the compatibility of this donor tissue with the host tissue. So that also a challenging thing. But another thing is autologous artificial liver products. That there have been few attempts to produce this autologous artificial liver, where uh, starting from patient's own cell, if we can generate the liver tissue or not. But main problem or main challenge of doing that is the primary hepatocytes. When we try to culture them for extended period, that is very difficult. So the primary hepatocyte, they cannot be cultured outside the body in vitro for a very long time, because they lose their phenotype and genotype, and they lose their function if we culture them in the body for a very long time. So that's why uh, there are several tissue engineering strategies people have been using for liver tissue, by providing, either by using, because as 2D culture is two-dimensional expansion of liver at the primary liver cells like hepatocytes is challenging. So people have devised various ways like sandwich culture, and also incorporating the cells in certain hydrogels to uh, enhance the time of culture, also to preserve the functions and not phenotype. Uh, so a few of the points we will discuss here. What are the things that is most important while taking while doing this liver tissue engineering? First thing, if you see liver in the liver, there are like other tissues. Liver also composed of cellular component and acellular component, but in liver the cell component is very high, because the hepatocytes almost comprise almost 60% of the liver volume is occupied by the hepatocyte. This high cellular density is very high, and these cells have very high proliferation capacity. Hepatocytes. So we have to always remember that if we are targeting liver tissue engineering, the cell density has to be very high. Other thing is that these cells are highly metabolically active, so they demand a lot of oxygen and nutrients. So those things we have to provide. Other thing, but as I mentioned, if we try to culture them outside the body, then they lose their phenotype and specific functions. So that is very challenging, and there, so we need to take care of this thing. But but it has been shown that the functional viability of the cells can be enhanced by when they form aggregations. Suppose if we culture them together, if they form aggregations, then this viability of these cells enhances. So that's the way by which probably we can devise our strategy for liver side cell culture. Even another study where by Stevens et al. What they have described, that in situ expansion of this liver cells is possible to engineer liver by by time in that form of aggregation. If we if we induce aggregation of these cells and if we then culture, then the viability of these cells enhances over, and for longer duration we can culture them. Like, so that's why they have fabricated a structurally organized tissue units and they have used this fibrinogen hydrogel. Fibrinogen hydrogel. Fibrinogen is present in our body. So it's a like it can be a autologous material also. So this secreted fibrin hydrogel, and they have incorporated human hepatocytes and fibroblasts. So these two cells they have incorporated, and then they have, so that's the aggregate. They form aggregates. That aggregates again engineered with endothelial cords. So they have used endothelial cells to make cords, and to which in these cords these hepatocytes and fibrous aggregates are connected. And then they have expanded it for some time, and then they have implanted in the mouse model of liver injury. And finally, they found that the graft, when they have implanted in the injured liver model, it expanded over 50 times through the course of 3 month study. And also it produced different liver proteins, that is, it's like the tissue specific proteins. So that is the function of that liver, suppose production of albumin and other proteins. Those things are, so this graft can produce this protein over the 3 month study for up to 50 times. So that's the important study, and by which they have shown the possibility of culturing or expansion of liver cells outside the body for longer duration, and further use of that thing for drug. Let us discuss a few more studies that is targeted towards this bioprinting of liver tissue. This is one such technique where uh, this Greek Sotel, they tried to print a liver model using that liver lobule-like structure, the hexagonal units, where this, if you see in the liver lobule, there are this blood vessels lining where coming from the terminal end to the central part. So this is from the terminal end to the central part, and these are lines, and hepatocytes are sitting on the sides of this blood vessels lining, because they get nutrition from this thing from the blood vessels, and as the blood passing through this channel, that particular that structure is called sinusoids, where endothelial cells are lined up in the luminal structure, in the luminal wall of this sinusoid, and then the hepatocytes. So in the hepatocytes, they get nutrition from the blood, and as the blood passes through these channels, the oxygen concentration in the blood that comes down, and so the oxygen at the inlet, the terminal end to the central end, oxygen concentration is high at the terminal end, and the central part where the oxygen concentration is low. So that this creates a zones within the liver. So like here the oxygen rich zone, and here it is oxygen deficient zone. So this process is called, because of this liver acinas, this forms acinas where different oxygen zones are created, and due to that the liver cells present in different zones, they perform different types of function. So in this particular work, they have tried to reproduce or recreate this liver lobular structures with DLP bioprinting. DLP bioprinting is a process by which this is where we have a bioink reservoir, where mostly this bioink is photosensitive material. We can load them in a reservoir, and then there is a printhead. That printhead is there, and the printhead can be lowered into this bioink, and then a DLP projection unit. This is a projection unit which project the whole layer by, it's also a layer by layer manufacturing technique, but this DLP projector, they project one whole layer at a time, and then when the whole layer is projected, that light is projected here, and first this printed will be lowered, and then the very fine layer of gap between this printed and the reservoir, that bottom part, and the DLP unit is on. So the DLP, and then it will project the light, that particular shape, the design of that particular layer, and then this layer will be solidified, and then after that, when suppose this is the projection, after that when this first layer is done, then this printed will be move up another layer. So there will be another layer, fine layer cap will be there. Then the next layer of the structure will be again projected here, and that will be printed that will be solidified, because this is a photosensitive material, and we are projecting light for to start initiate the photopolymerization. So by this way, the material again here the material will get solidified, and again after this the this the print head will move another up again for another layer, fabrication of another layer. This process keep repeat. This process is repeated over time to fabricate the whole 3D structure, and then it can be the printhead can be moved up, and this thing can be washed and further cured. Certain time further curing is required. Also, then washing and something is required to remove the uncured material here, because uncured material can be toxic. So uncured material is removed, and then the cured or photopolymerized material structure based upon your design is ready for further use. So that's what we had explained. This is the structure, this is their design CAD model, the model of this of the structure, and this is the printed structures. See here, these are the channels, those are open, and the rest of the structures is like this. So digital light processing projection, DLP, so is also a layer by layer manufacturing process. As I said, after each layer, the printed move upward in the next layer, and each layer was got polymerized onto the previous layers. So this uh, layers they get bonded by this during polymerization. They get bonded, and by this thing, the layer to layer integrity is maintained. So after complete printing, the formed tissue model is removed, printed, and ready for cultivation. So that's what they have done. This is the structure they have printed with the morphology of the printed structure. You can clearly see microscopic 10x images there. This is 2x and 10x images they have shown. So the channels are also open. The channel dimension is almost like 191 micron. And because probably they have designed the channel dimension to 200 micron, and they get something like 190 to 210 micron channel. That ranges channels they have get. After clear visual, after complete visualization, the channel is hollow. They have visualized the material, and the channel is hollow here, and the channel bottom is very clear. See, also the channel is open. That is, the channel structure is hollow by infusing trypan blue, flushing the trypan blue through this from the central part. They have shown that the channels, you can clearly see visible channels are quite channels are open. So this structure they have shown. They have also done various other studies, like they have when they encapsulated this when they encapsulated hydrogel hepatocytes within the hydrogel and put into this thing, so the cell viability was maintained, and then they have also seen that the production of liver specific proteins are also present, also there, and also they have seen that different tissue specific gene expression on this study. So these are the studies that mostly after bioprinting people do. First thing, they observe the cell viability within the construct, because they are printing or printing whether the cell viability is maintained or not. Other thing people generally consider is the tissue specific gene expressions, like the cells, the encapsulated cells or the printed cells in the structure, whether they express a particular tissue specific genes or not, and also the liver specific functions like albumin production, urea production. All these things are also people check, researchers generally evaluate after bioprinting process. Similarly, there are several methods people have used, or different 3D bioprinting methods have been used. Methods have been used for fabrication of liver tissue constructs using different types of hydrogels, and also, so this is one such method where scaffold-free 3D bioprinting strategy was developed, and this method was developed by Professor Koichi Nakayama from Saga University. So in this method, basically it relies on cellular self-assembly, where suppose if you put the cells, either it can be homogeneous or heterogeneous cell types. So these cells, when you bring these cells, like you put them together, the cells have that intelligence. They adhere to each other, they assemble, and then they produce a a produce a agglomerate. That agglomerate, then they produce this agglomerate. The cells, they present in this agglomerate, they produce extracellular matrix components, and those extracellular matrix components actually that bind the cells together, and then a tissue structure as tissue structure is developed. So in this process, now this process is also very similar to the spheroids or organoid preparation, but here the this but this group has done, they have first prepared these spheroids, and these spheroids, this like round, the circular or the circular structures or spherical structures, these spherical structures were then were assembled by means of a array, micro needle array, that they called Kenjan. So with this, suppose these spheroids are prepared, on this these spheroids are put into this needle and stacked inside the needle in this micro array, in the micro needle array, and then they allow it to self assemble. Now when we put them very close to each other spheroids, then they assemble get assembled with each other, and the cells can actually migrate, and then they form a complete continuous tissue like structure. Now depending upon the microarray, depending upon the microneedle array, we can get different types of structures. Either we can get a circular, we can get suppose we get cylindrical structures, we can also get a maybe a square or cuboid structures. So different types of structures can be possible by designing this microneedle array. And then also there is a stage here, so that can be after fully formation of this whole structure, then that can be lifted up and you can get this structure. So now this is here, this is material free technique where no biomaterials have been used, but it relies on the cellular self assembly, and then we can get the self freestanding structures. So this method is called Kenjan method for of scaffold free 3D bioprinting technique. Now this method has also been employed for fabrication of liver tissue construct, liver bud, basically liver tissue construct for further transplantation or implantation. Like this is in this case what they have done in this particular paper, they have used three different types of cells like human hepatocytes, HUVEC as human umbilical vein endothelial cells, derived endothelial cells, and human MSCs, mesenchymal stem cells. So these three cells they have combined together and put them in a U-bottom 96 well plates with a low attachment surface. Suppose this U-bottom 96 well plate, they have very low surface, they have very low attachment surface area. So cells generally they don't attach to the plate, rather the cells they start assembling with each other and then they form spheroids, or in this case they call it liver bud like spheroids, right? So this thing, so by combining these three cells together in a U bottom plate, then they come up with this liver bud like structures. Now these liver bud like bud spheroids, these spheroids, then this is without manual, there's no manually, it's automatically through the they have fabricated a bioprinter, designed a bioprinter for to suit this process, where this robotically these spheroids are lifted and then place them on this microneedle array, as I have discussed before. But as per the design, suppose in this case they want to make this kind of cuboid structure or a rectangular structures. So they arrange this microneedle in such a way to suit this fabrication process, and then these spheroids are put into this needle array, microneedle array in that particular order. Now then upon culturing for some time, then what will happen, the cells present in the spheroids, they will assemble with each other, they will migrate, they will self assembly will happen, and finally a continuous tissue will get developed like this, and then that can be used for transplantation onto in the parenchyma of liver. So this is the strategy they have been employed, just as the diagram shows here, and this is for a tissue transplantation strategy. And scalable liver tissue construct, scalable liver-like tissue was constructed in this case. This case, the liver-like tissue is really, it's not small. It's a good size tissue, a good volume of tissue. Then they have, so this is another thing how they have, there's the further, say, a few more images showing the further details of this work. So where they have constructed a scalable liver like tissue. So both these strategies they have used spacing and without spacing. When it is spacing is not given, then it is a continuous tubule like structures. But when a spacing is given, then it can be a porous porous tubulous tissue like structure. So here you can see here clearly 3D printing. During 3D printing, here see the micro needles are visible here, right? So where the spheroids are placed on the micro needle array in that particular order, and then when the after removal from the needle, still we can see this needle space where the needle the space is visible after removing that construct from the needle. And then when it is cultured for some time, then you see these pores are no longer visible, but here it is a continuous tissue like structure is clearly visible. So this here, this is construct is nothing but the cells, all the cells are there, and also cell plus hydrogel, no, not hydrogel, sorry, cell plus cell derived extracellular matrix is present. So it's taken. So this is the top view of this construct, and this is the side view of the same thing. Where you see in this case, the some spacing is given in between the each spheroids. Then some here some amount of porous structure can be clearly seen here, and the tissue is also little bit porous structure. So you see here, the scale bar is 200 micron. So it is just all the tissue is almost like 2 to 3 mm of size. Right? So this is the method they have used for tissue. And then they have done transplanted this thing in the parenchyma of liver. What they have done actually, they have ultrasonically, they have burnt a part of the liver here. So that part is get charred, and you see this tissue is after this ultrasonically activated device, this tissue here, the transplant bed, that was necrotic and that degenerated into a yellow like tissue. So they have removed a part of that thing, and they put their structure, put the bioprinted liver in that case. What they have fabricated, that thing they have put it here, and then they cultured, and then they allowed it to grow for some time. And this is the histological section where you can see clearly this area. So here, this black dotted area there where the tissue is actually implanted, bioprinted tissue is implanted. And if you see this area where this dotted area, that is the implanted tissue, and there is a native tissue. So there is not much difference, and tissue can well integrate with the surrounding native liver tissue. And this is the thing that by fluorescence assay they can see the implanted liver tissue. The engraftment can be clearly visible upon with days. This is the again at the end they have shown the bioluminescence as imaging of the liver chip transparent fat surface of here. And here you can see that the transplanted liver tissue can be integrated with the thing, but in certain liver chip, this is the liver chip and this is the native liver, where where this part was actually necrosed by ultrasonication burn. That part you can clearly see the vascular occlusion was clearly observed here. The vascular occlusion because this part was burnt, and also the and the transplanted liver chip was packed with clots, so that can be clearly visible. So here you see here, this is the part was with the transplanted clots were packed with brown clots, but the liver chip or the bioprinted liver model, the bioprinted liver tissue, that was doing fine, that was well integrated. So here in this paper they have shown a a scaffold-free 3D bioprinting technique for fabrication of these liver tissue constructs. And also they have another thing they have shown here is the engraftment of the bioprinted tissue and well integration and functional tissue regeneration with the bioprinted liver model, liver tissues. So this is a very important study where they have shown the possibility of generating liver tissue with this bioprinting concept. Now, there are various other works also describing different uh, or targeting different tissue structures of the liver. Like some people in some research, they have targeted the liver lobule-like structures, and also some research that also multilobular structures have been also been targeted. In this particular work that we are going to discuss now, this group, Lewis et al., what they have done, they have prepared a intrahepatic biliary tree. You see, in the liver, the two different circulation system. One is the that blood vessels coming from the portal. There's a portal triad, but the portal vein and also the liver artery supplying to the liver and bile duct. All these things that form a portal triad. From there, that circulated through the liver lobule and going to the central artery. So from there, this is that pass through. So the bile duct is a separate circulation system, and then the bile duct is generally present. Bile is mainly produced by the apical surface of the hepatocytes. When the two hepatocytes they touch each other, they are connected to each other. That from the hepatocyte, from there the liver, the bile is produced, and then it collected. It gets collected in a bile duct, and in the bile duct where cholangiocytes basically they are present in the bile duct where they are the thing, and cholangiocytes they maintain this means also by this way the bile is getting circulated from the liver to the for for they again it helps in digestion. So in this particular work they have used a sacrificial material, that is plurionic 1 F1 to 6 1 to 7, and plurionic, as we have already earlier discussed, is this material has been widely used as a sacrificial material because it has a property of it's a thermoreversible material. It's above 20 degrees centigrade, so it behaves like a gel-like material, so we can print with that material. But and again, if we reduce the temperature below 20 degrees centigrade, then it will form a gel. It will form a it forms a liquid. It behaves like liquid material. So we can that time we can take it. So the idea is the strategy is mostly they print it above 20 degree at room temperature like 25 or so, and then then a template was created with the sacrificial material, and then the other material was either can be poured or can be printed in the same structure. Then when the complete structure is cross-linked, then the temperature can be dropped down below 20 degrees centigrade, and the plurionics, when the plurionic become liquid, and then it can be removed. So by this strategy has been used in several research paper to generate vasculature, to generate vascularized tissue, all these things. In this case, they have used to develop a biliary tree. They have used a similar concept where they have also used DCM hydrogel, that is liver tissue specific hydrogel, as I have discussed in my earlier lectures also. That tissue specific DCM, like tissue specific extracellular matrix, it has a very significance in tissue engineering because it can provide a natural biochemical, biophysical microenvironment for the resident cells. So the cellular growth function that can be improved further. So in this case also they have used a liver tissue specific liver tissue hydrogel visualized, where the cells are gone, and then that is extracellular matrix or acellular matrix that is present, that can be developed into a hydrogel. So in this case, they have seen the effect of this DCM for engineering the intrahepatic biliary tree by encapsulating in biliary epithelial cells. Mostly these are cholangiocytes. They have been encapsulated those things within the liver DCM, and then they have shown that this can lead to the formation of complex biliary trees in vitro. And also they have also worked with the geometry of these structures, and they're using by designing proper biliary tree with the help of DCM. and using pruning, they have shown that a biliary tree can be functional, can be formed, and then that can be used. So in this work, the aspect, several aspects have been very by, like DCM structures, geometry, width, angle, all by optimizing all these things, they have formed a biliary tree-like structures. Now this biliary tree has, as I mentioned, this is very important for liver specifically liver function, because bile production is the one of the major function of the liver. So now, as I have seen the earlier work, like the formation of the lobule in one typical work, lobule-like structures, and then this biliary, and then the other whole thing. So if we now all these are as can be used as a building block for further development of this liver tissue constructs. Like, so the challenges for liver bioprinting is the because liver microvascular network is a highly complex vascular network. Liver has this, and there it is in the micro scale first thing, and then it has a network. That, so we need to create that kind of network. So that is the main challenge of liver bioprinting. We need to create that kind of complexity. Other thing is this reproduction of this liver acinus structure, because liver acinus, as I mentioned earlier, is the functional unit of this liver, and the function of the liver, like detoxification, metabolism, all this depends on this liver jonation or liver acinus structures. So reproduction of that thing is very much required, and also it is highly challenging. That is another major challenge. Other thing is the scaling up of the individual structures. We have discussed in this class the different individual structures like this hepatic biliary tree, or liver acinus structures, or even the liver lobules like structures. So all these things, the scaling of is another major challenge. How we can actually scale this individual units? You can combine them and you can scale them up to come up with a fully functional liver tissue. That is another challenge. Other thing is the till now people have shown these individual structures have been done, but when these things are connected, and if that is suppose that is implanted in the injured liver model, so that the anastomosis with bile duct, portal vein, hepatic artery, that is still not been realized, because it is not being done, because unless suppose we have bioprinted a structure, unless we connected that, suppose that we printed in the bioprinted structures, we have bile duct, we have portal vein, we have hepatic, all these things are there, provisions are there. Now this thing has to be connected with the pre-existing bile duct, that the bile duct with the pre-existing one, and the similarly portal vein with the pre-existing portal vein, and the hepatic artery with the pre-existing hepatic artery. So unless it is done, the circulation cannot be established, cannot be realized. So this thing has not been done. So these are the challenges that researchers have been working on for further development in this liver tissue bioprinting. So thank you very much for attending this lecture. We'll see another tissue bioprinting in the next lecture.